Okay, good afternoon again to the last event of this nice meeting. Björn will say a few words to wrap it up after this. So I'm just going to first say that I'm not going to introduce Rudy, someone else will do that. But Rudy has a connection to polyelectrolytes and electrostatics, and I think he will say something himself about it. He talked about it 3 years ago, but it's an interesting story. So Rudy is here not only for this talk at this workshop, but there is also a symposium next week on photosynthesis, and that symposium is actually in the honor of the introducer for Rudy, whom I will present now, and that is Professor Bertil Andersson. So Bertil Andersson, some of you know him since 3 years ago. He just stepped down as the president of NTU. He came here from European Science Foundation. Of course, he's a plant biologist with 300 plus publications and so on. But came here 2007 as the provost and became the president of NTU in 2011. And of course, in this time, NTU has become the fastest rising research-intensive university in the world. And much credit goes to Bertil for driving this and pushing all of us at this university to achieve these heights. So, of course, Bertil is a close friend to Rudy and knows his work very well, and so it's very nice to have you here to introduce Rudy Marcus to give his final talk here. So please, Bertil.
Thank you, Lars. Yeah, I've just retired, so I'm a free man, you know, it's really nice. So, but I'd like to have a few duties like this one, so it's really an honor to this afternoon introduce the next speaker. I guess you already know who that is, so it's not a surprise. And you always say that you should save the best until the last, and I think we are doing this at this very conference here now. So I'm introducing Rudolph Marcus, and if I will give some affiliation of him, I will of course start by saying that he's a visiting professor at Nanyang Technological University. He's even an honorary doctor at this very famous university. Of course, maybe, to be fair, say he's also the Noyes Professor of Chemistry at Caltech, but yeah, here in Singapore that is of less importance. I also have a Swedish connection with respect to Rudy. He actually received a modest prize back in Stockholm some years ago. That's called the Nobel Prize, and that was in 1992. He received the Nobel Prize, and I guess you know for what? For his contributions to the theory of electron transfer reactions. In other words, you could say Rudy Marcus got the Nobel Prize for Marcus theories. I think that was very appropriate, and of course we worked a lot on that in the committee, because I belonged to the Nobel Prize committee when Rudy got the prize. So that was a very interesting work. And of course I remember him coming to Stockholm in the middle of the December darkness, but despite of the darkness and the coldness, he looked quite happy. So that was, I think, it was nice. So Rudy, as you probably know, is not exactly a rookie. His PhD is from McGill in Canada, and that dates back to 1946. I'm a retired guy, as I say, but I was not born when he made his PhD, so that sets some perspectives to things. And he's also been, look in the back mirror, professor at University of Illinois, but I think since 1978, Rudy has been at Caltech. Of course, his work is very seminal. We're going to hear about it, and it's been honored by the Nobel Prize and a few other prizes as well, I would argue, and Wolf Prize, for example. And 400 papers plus. Of course, electron transfer, I'm just, as Lars pointed, a simple-minded biologist. working in plant research and photosynthesis, but of course for us studying photosynthetic light-driven electron transport, the Marcus theory was very important, and also in other areas of life science, as well as chemistry, physics, and engineering, I would say. So this has broad impact. So we are indeed happy that Rudy is here with us here today to give at this conference and at NTU and giving the final talk. And it has the short and snatchy title, Theoretical Analysis of Complex Systems and the Role of Phenomenology and Computer-Based Calculations in Treating the Experimental Data. What about that? Rudy, please.
Well, it's a real pleasure to be here and to have this opportunity of seeing many old friends. Oh, wait a minute, I guess I have it here. And to have this opportunity to recall some of the work in polyelectrolytes briefly, and also what we've been doing much more recently. My ventures with polyelectrolytes was a very brief 2-year period. I published papers in it in 1954 and 1955. They were papers that arose because of a question that a graduate student asked. I was at Brooklyn Poly teaching at the time, and Brooklyn Poly was a polymer center, and polyelectrolytes was the subject of much research. And this student asked a particular question in some course that I taught, and I thought about it, and I thought, well, boy, that's an interesting question. So I started to read about polyelectrolytes a lot, and read every book that Brooklyn Poly had on, or at least looked at them on electrostatics to see which one would provide me with the deepest insight, and I found one of them that did, and then ended up writing those 2 papers. So that was 54 to 55. And then in what the advantage of the book was that I looked at, and it was Mason and Weaver actually, was that it was so physical. It wasn't just a matter of a collection of a few formulas, but they really went into what each symbol meant. So, and then in 1956, I heard about some unusual problem where Bill Libby had applied the Frank-Condon principle to an electron transfer reaction and got very excited about it because normally the Frank-Condon principle is applied to spectroscopy and not to chemical reactions. But here is a matter of the electron jumped in, during the time of that jump, there wasn't time for the nuclear to move. In other words, the Frank-Condon principle. So I was very excited about it. And then, but the same day, after a while, I became very uneasy. It was sometimes when something is wrong, you may not immediately identify it, but you may feel uneasy. Anyways, I did, and then I realized that as great as Libby's idea was, it violated the law of conservation of energy. Well, I don't know if things have changed in the intervening years, but in those days that was not a good thing to do. So the question was, how could you invoke the Frank-Condon principle and not violate that? And then I realized that, well, one had to have fluctuations to occur to produce a sort of unstable states, and then you could make that happen. So that was, to do so, it was a matter of taking what I learned about the electrostatics and polyelectrolytes and going one step further and dealing with non-equilibrium situations, fluctuations, but heavily based on what the polyelectrolytes told me. And then within a relatively short time, developed the electron transfer theory. In fact, in that work, unlike any other work that I did, in those days I worked hard, in that work, unlike any other work that I did, it took 1 month to get the electron transfer equation. I mean, that's, maybe it was my youth that made me work like that. So that was my brief sojourn with polyelectrolytes, but for me it was a very important step because without that background I don't think I would have been able to do anything. All right. What I'll do is say something about electron transfer reactions and a little bit about the theory involved, but more on the way one applies the theory to problems. When you have complicated problems, one way is to try to solve everything completely, so you know, that, for example, the rate of everything. You've calculated everything. But it may turn out, although you've calculated everything, you know nothing. All you've got is numerical results and so on. And besides, that's a lot of work. You may not have learned anything about relationships between various kinds of processes and so on. So one of the themes that I'll be focusing on is what can one do in terms of developing relationships that one can test without any calculation. You relate A to B, you measure A, you measure B, and you see if they're equal or not equal. So, and that theme will carry over to more complex systems that I'll talk about involving a molecular motor. All right, here's my one polyelectrolyte slide. You may not recognize it as being a polyelectrolyte, but this is what it looked like back in the 54 and 55. And so, as I mentioned, I studied a lot about electrostatic properties. So now, let me say briefly, because it's not the main focus, something about electron transfer reactions. During the Second World War, there was lots of isotopes effects studied, and so lots of different isotopes were available, and people could measure these what's called self-exchange reactions. The asterisk indicates a radioactive isotope, so it's used as a tracer. So they could measure what was the simplest class of reactions in all of chemistry. That was what helped the theory, actually. It was a reaction in which no bonds were broken or formed. That's a big simplification, just an electron jumps. So by means of the radioactivity, the reaction would occur. They were able to separate the two kinds of compounds. Here radioactivity has appeared in the other valence state. It started off in that valence state. It's only the electron that's moved, not the radioactive atom. So these are examples. And because of the simplicity of these systems, it was possible to sort of derive some generalizations which led to Bill Libby postulating the Frank-Condon principle. So it was the nature of the data themselves and the simple nature, some aspect of it, that forced his wise application of the electron transfer system. This briefly is the theory. It's an example. These are cartoons. These triangles are intended to represent ordered water molecules. These are ions of +2 and +3. The +2 is a little bit bigger. This is an exaggerated thing, and because they're extra electrons and so on. And generally, the molecules are more oriented around the +3 than they are around the +2. After an electron transfer, it's the reverse. So somehow you have to change those orientations to those orientations, and you do so via an intermediate state that which the idea is to calculate its properties and so on. And that we did with dielectric continuum theory and so on. But essentially, the basic idea is that for an electron to transfer, it couldn't just transfer. That would violate energy conservation. So you had to have changes going around in the vicinity of each ion. And on working through the theory, it led that you could take these 10 to the 23rd atoms that are in any system, and you could represent things eventually by free energy curves. And that was a big visual simplification. And here are the reactants. They turned out to be harmonic in the type of approximation I was using. Computer calculations many years afterwards showed, in fact, they were indeed harmonic or close to it. And here's the products. And the idea had to have fluctuations from there to reach some place where those two free energies are equal. Actually, energies are equal, energies are equal. And then you could get electron transfer. So this type of diagram came into the process. And this was the expression for how fast the reaction was in terms of the driving force of the reaction, and something that we introduced we call lambda, which is about those changes in bond lengths and reorientation of the water molecules and what have you. And this was the formula that was developed in the 1956 paper. This lambda depends on the sizes of the ions, on the difference between the optical and the dielectric constant, because in the response of the water to the charges, it's difficult. It's not the electronic part of the water that's the problem. It's the dipolar rearrangement, the nuclear part. So that takes care of that. And these are for the inner shell thing, force constants and square change of coordinates. Anyways, the main thing is that it was possible to do something. But now we come to the key point, the point which will carry over to other kinds of problems. It's one thing to derive an expression, and here you are. And then I was able to show that if you consider a cross reaction, I'll call it 1, 2, you could relate the reorganization, that lambda, to those of the individual isotopic exchange reaction this way, and that produced this relationship of rate constants. In other words, if you take two reactants, and in one case study the isotopic exchange of it in its oxidant form, and in the other case study the isotopic exchange of it in its reductant form, the reaction itself rate constant is K12, the isotopic exchanges are K1, etc., and this is an equilibrium constant related to standard free energy reaction, and this is a function which is usually close to unity, but in terms of those properties. So here's something that one can measure the various quantities. One can measure the self-exchange rate there, the self-exchange rate there, the equilibrium constant, and one can measure that too. And one can predict the cross reaction. And then one can test that to see if it works or not. In other words, here's something, no computers, just comparison of experimental results and see if they agree or not. And this will be a theme that I'll carry on to some more complicated systems. So this was sort of one of the key developments of the theory, because it permits you to apply the theory without having to do a lot of calculations and so on. The other things are important too, but this had that advantage. All right, here comes the surprise to me. I derived that formula for simple electron transfer reactions. Well, you know, sometimes experimentalists are even more daring than the theoreticians, maybe usually more daring than theoreticians. And one of the leaders in the field, Norman Sutton, applied that relationship that I showed you not to an electron transfer reaction, but to an atom transfer reaction. In other words, he knew the K's, little k's, he knew the capital K, he could predict the cross thing, and it worked. So here is a relation derived for one thing, and it worked for something else. Well, then experiments were done much later. Here, this is, what did I do this one? Oh yeah, I did this. I'll give the calculations first. Here's some people who did some very elaborate quantum mechanical calculations. And so they got their K's by all capital K by quantum mechanical calculations. And here that simple quadratic formula worked perfectly. This is the theoretical line, and those points are the experimental points. So it worked very well. So here is a relation derived for electron transfer that worked for something else. And this will come into some other more complicated systems. These were experimental results for this reaction. This is a transfer of a methyl cation from one aryl sulfonate to another. And once again, this is the calculated, there are no adjustable parameters there, and the agreement is beautiful. In other words, the central theme here that we'll try to repeat for more complex systems is that sometimes if you have a theory, you can make predictions that you can test without any calculation. Probably doesn't happen often, but it happens occasionally. I will show you another example for a more complicated system. Well, this reaction and the one I just showed you before, the theoretical calculation, were not electron transfers. They were methyl cation transfers. And so back in 1968, I decided, well, let me try to treat a group transfer thing and make some calculations with some crude model related to what Pauling had developed for something, bond order, bond energy model. And we got something about the barrier. It's not exactly a quadratic term, it's something like that. But nevertheless, it's approximated by that quadratic formula. So this should be the formula that people would be applying to those methyl cation transfers. But its results are very similar to what the quadratic thing. So even though the two classes of reactions obey something similar when you compare cross reactions with the self-exchanges. There are subtle differences, but nevertheless, the simple formula works well. So this made it possible to understand why applying the simple formula to something for which it wasn't designed actually worked. Let me give another example of use of a totally different example now. It's a total change of pace. Of a different example of something. Here's a system, a well-known system, titanium dioxide, okay? Used a lot, solar energy conversion, what have you, here and there. It's an ugly looking thing, isn't it? Just ugly. Imagine somebody trying to do theoretical work on that. I mean, how stupid could they be? You know, or they must be living in a different world. So anyways, this is titanium dioxide. And so one of my students, and I take just very little responsibility for it, I mean, but I should take more, but it worked well. So anyways, I suggested to him, well, let's apply a very simple theory, a diffusion theory, for the process that's involved, where you have a dye on this terribly complicated thing, and photoexcite, inject an electron, watch it return. You can watch it return by fluorescence behavior. And let's develop a theory, a very simple kind of theory, pretending that the titanium dioxide is a flat thing, pretending that the dye is like a circle, you know, and we could apply very simple formulas and so on, and we got some sort of a power law for the return. And here's an example of I mean, these are some experimental data obtained by Ed Yao here at NTU in 2009, and this is the theoretical calculation for the slope. The slopes agree very well. It's what's called a power law. This is a recombination law that's not first order or second order, but because of a slow diffusion, it turns out to be a power law. So it's an example of taking a complicated system and making a simplification in the analysis of it. In other words, when you treat an experimental system, there may be some features that you can approximate by idealized thing and have it work and so on. All right, let me turn to the next example, which will be the final example of this ATP synthase, for which a Nobel Prize has been won. And this is an enzyme which functions to pump hydrogen ions and other ions, cations across the membrane, and in the process it is an diphosphate inorganic produce ATP, or you can run it in reverse and drive and sort of drive ions in the opposite way. So here's sort of this is a structure on the top for which the prize was received, and now they have something about this structure too from the cryo-EM that's been obtained. And if you look at how does nature sort of do this molecular motor? By having an ion gradient, and somehow by having offset channels of going in and then coming out the other end. Offset channels use an acid-base reaction to actually pull a rotor around, and in the process make this asymmetric rotor do all sorts of things to the things on top of it that permit these subunits to open and close. I mean, it's absolute, ingenious that what nature has done. So let's look at some experimental data related to it. This is an example of a cross-section. It's a cartoon. This is a cross-section of this rotor that as it turns asymmetrically affects these things, causing them to open and close. For example, an ATP can go in, and then with the rotor turning, it can close it. That can force an hydrolysis, and that can eventually cause the ATP in another place to come out, and then the inorganic phosphate to come out. In other words, I mean, what nature has put together is great. All right, well, what kind of research can be done with that? Well, there's been a lot of single molecule experiments that have pervaded many areas, and here is what it's done in this field. there are experiments which you can take the top of that motor, the part that's actually out in the subunit, out in the water, not the part that's in memory. You take that part, and you can sort of cause it to turn, and you could stall it at various angles by magnetic tweezers. You can stall it at various angles, and you can hold it for various lengths of time, and either it'll go forward or go back. From those kind of data, you can get rate constants. So that's one kind of experiment. Another kind of experiment is that you can control the rotation of that central unit, that gamma subunit, and that will cause things to open and close and ATP to react and what have you. So you can get data on that. And you can tell in those data whether an ATP is inside or outside. Because if it's inside, if you've put a fluorescent attachment on it, it'll fluoresce. When that same species is outside, it won't fluoresce. So you can follow this thing. Fluorescence is commonly used, as you know, in this thing. So you can do these controlled rotation experiments. You can do experiments where the thing freely rotates, and by fluorescent techniques and or observation by optical methods, you can see something about its rate of rotation. So you can do all that, and now here at NTU, there's some work done in a much faster time scale by yet another method. So what about, so now we're dealing with a more complex system. So what can we do there to possibly use some data to predict other data without any adjustable parameters? And of course, you may have a model to do that. And if you're successful, then that's good. If you're unsuccessful, then you step back and you have to make a different model. But let's see how much one particular model that used. Okay, here's an example of, oh good, the animation seems to be working. No, yeah, see, this thing is here rotating, it's freely rotating. You can see there's something which you can see it fluorescing and so on in spurts. And here's an example of the data that comes out of it, the rotation angle versus time as inferred from this optical data. And you can see that it's moving in steps. This sort of gave direct observation of the idea that the motor moves in steps, so on, and not just continually rotates. And you learn something from the steps. You learn about rates and what have you. So it's an example of one of these single molecule techniques that give you data that you could never have obtained in the older days. Here's an example of where you do instead a controlled rotation. This is changing the angular velocity at a constant rate. And here is the fluorescent moiety. It's fluorescent when an ATP or an ADP is inside, and is not fluorescent when it's outside. These are worked at low concentrations, so you distinguish that. And so you see there's fluctuations in there, and that's more such data. So this is an example of what you can learn at the single molecule data that you can eventually hope to translate into understanding of what's going on. All right, what about the theory? Well, here are those curves once again. This is rounded off now because it's not an electron transfer. And essentially, in one position where something is open, you may be here with the reactance being stable, and as it rotates, you may end up with something being closed and the products being stable. The main thing is that during the rotation, you have free energy changes. So somehow, to develop a theory of this mechanical chemical motion, you have to have an expression which combines both that motion and the chemistry. That's the essence of the theory. And so what we did, forget about the equations, that's what it is. This is this free energy barrier. You may see this lambda, this quantity that appeared first in electron transfers and also applied, unsurprisingly, to atom transfers and so on. Now we're applying it to a different type of thing. These are various work terms. The main thing is this, that part of the free energy expression are these things that depend upon the rotational angle. So in this expression, we have things depend on the chemistry, things depend upon the rotational angle. The details don't matter at all, of course. It's just what you put into it, that you've got both things present. And we're doing it, we're combining the chemistry with the mechanical motion through a free energy expression. These are torsional motions, both in the starting position and the final position, because there are different stall angles that were shown up in the free single molecule experiments. So here's an example of the result. The experimental points, this is for the rate in one direction. The blue line is for the rate in the forward direction. That happens to be binding of ATP. The red is for the opposite. That's for the ATP coming out before it's had a chance to do anything else. And this is the equilibrium constant here. Those experimental points are the dots. The theoretical curves in which we use other data, not these data, but other data to make the calculations, to evaluate the things in the theoretical expression, are the straight lines. So that type of agreement, no adjustable parameters, okay? So what it means is that the model at least is capturing a central feature. To us, that's important because we're now working on some other problem where our first effort at doing that, it didn't capture. It was wrong by a factor of 2 and an exponential and so on. So you can get wrong things, you know, so you have to explore. This time it actually worked first time round. The co-workers, Sander Volken-Katsko, and we published a number of papers related to this, on this. So here's an example of what is truly a complex system, and where one introduces approximate ideas, maybe based on simpler systems. I mean, the essence is how do you combine the chemical and the mechanical motion? And we did that in a certain way. And you use it as a vehicle, not for calculating everything a priori. That'd be a separate problem, which people could try to do by computers, but for relating things. The same thing, in a sense, that was involved in relating those cross-rate constants, the K12s to the individual isotopic exchange. Again, not detailed calculations, but having a relationship. And that's what we have here. So this is a summary of what I just said there. There's a motor, generates the catalysis. You have a catalysis. We broke up the catalysis into steps. We had a free energy equation for each step. We predicted and test relationships. We turned different kinds of single molecule experiments. And now what I think is an interesting point, here's this analysis. So now what we're trying to do, we being some coworkers, is now do some computer calculations guided by this information. In other words, now it's computer calculations, not saying, well, let's start everything from scratch and calculate, but let's use the knowledge that we have with specific goals in mind, restrict the computer problem so much, because there's so many unknowns, so many things, so many places where things can go wrong. The parameter space is too big, and so on. So when one can do it, and there are probably many cases where one can't, this is an effort at introducing approximate relationships that make predictions relating experimental data, and when those predictions are verified, to use it as a guide in then making some computer calculations that we normally wouldn't have done without thinking about this. Computer must be telling me it's time to stop. Well, anyways, this is an example of what we were trying to bring out. That with the computers, I mean, it's fantastic what they can do now that we couldn't do. And I've seen that in so many cases. I remember in the early days of unimolecular reaction rate theory in the gas phase, in which I developed a theory, it's still used, RKM. and how little we knew about absolute values of things, and how for those relatively simple systems, one can calculate things now that one never dreamed of using computers. So computers have been, and this is one little example, have been fantastic help. But now the question is, as we move to more and more complicated systems, to what extent can we strive to have some sort of a model that sort of provides relationships to tests? Then having done that, focus the computer to calculate and test various things, what have you. Well, it's always a pleasure to come back here to NTU. I've had many exciting times here, and I hope to have many more. Thank you.
Yeah, so first, Rudy, thank you very much for this wonderful lecture, and yeah, I think it's a wonderful learning pedagogical lecture for the young people in the audience, the future scientists, on how to think creatively, yet at the same time simply in science, and not necessarily complicate things too much, which we often do, all of us, particularly when we have these wonderful computers, and it's kind of tempting to just brute force all atom or even quantum included, complicate things. But anyway, if there are questions, of course, I'm sure Rudy will be happy to take them. Yes, so Gero has the first question.
I have a rather simple one. You gave
those are the difficult ones.
Well, you made a major point, of course, in finding relationships and establishing relationships.
Right.
And you said that actually the parameter space is so big.
That's right.
Now, if I look at that from a neutral viewpoint, it seems that it takes people with specific skills to actually be able to see that relationship or to see the beginning of this relationship in the first place. So it is a rather personal question that I would like to ask, and the question is, do you think that you can actually train young people to get these skills, or do you think that it's more of an individual thing and that you have to foster your own talent and see how that turns out?
Well, of course, I've had many students, and they've ranged in their intrinsic ability. And there are some that right at the very beginning showed great promise and continued to do that later. And there are others which showed great promise, but in a much more mechanical way. It's not often a focus of people to do that. So it may require some intrinsic thing, on one's part, just that kind of curiosity. I'm not sure if that can be taught. I mean, one could do something by example, yes. And people sometimes, the students have sometimes told me what they've learned, and this and that. But as I look at things, I think that there has to be something inside that it's part of one's life. That one is looking for analogies without even thinking about it, in relationships and so on. But I'm not sure.
Okay, thank you very much.
Yeah.
More questions?
Yes, thank you very much for the lecture. So I have a general question. I very much like and agree with the idea of a simple theory which can guide computation and guide research in general. My question is, how do you convince biologists that the simple theory that actually holds water, and then they should follow it, or at least take it seriously?
I wish I had successful experience in doing that. To convince anybody, of course, you can't do it on the basis of the beauty of it. If they didn't create it, it may not look beautiful to them. It has to be done if there's a real benefit to them. So, in a way, one might have to prime the pump there. I know, for example, in the electron transfer stuff that I did, there was a theory out there, and there was an immediate positive reaction to it. But in order to encourage the use of the theory, several years later, I think it was 1963, I then wrote a paper in which I applied the theory to a number of different kinds of examples. And that was sort of like priming the pump. It's always better if other people prime the pump, but that was so one can try to do that by examples so that one sees the relationship. Because for somebody from a more sort of down the road applied area or something like that, to use it, they have to learn it, they have to be convinced that it's worthwhile learning. And this way, if they see an example of where it's been used, then that may encourage them. So it's not just something, you can't just put it out there, I guess. Occasionally you can put it out there and have it used. Indeed, in a different field, what's called unimolecular reaction theory, I did that in 1992, boy, I've forgotten the years, 1952. I developed what's called RKM theory, and it still is a standard theory, the standard theory of its choice in field of gas phase unimolecular reactions. Well, there I didn't prime the pump. There were practically no data available at that time. And so it wasn't until 8 years later, around 1960, when data had accumulated, and other people happened to have seen the theory and thought, oh, well, let's try it, you know. But that was an example of not priming the pump. Of course, there weren't data to prime it with. And it took 8 years, whereas in the electron transfer theory, once the pump had been primed, I think it was immediate response. So to some extent, one has to go to work on one, you know, to go through all the nitty-gritty of applying it to actual systems so that people who are less trained in it can see, well, there's something to it. Like, for example, I remember Graham Fleming at Berkeley sending one of his graduate students to Shaul Mukamel at the University of Rochester at the time, because Shaul had developed a very sophisticated theory. It was very difficult to understand. Shaul was absolutely brilliant, but this theory was very difficult. So Graham sent a student to spend 1 month there so he would learn enough to be able to apply the theory. So sometimes a lot of work is involved in applying a theory. You know. But it's a very good question you raise. You know. How do you get things rolling? I've seen this in connection with some other stuff that other people have done, where somebody was telling him about some work that he feels is the best way of doing it. And a few people here or there praised him for doing it. But I think if he wants to get that well known and well used, he may have to do more pump priming or persuade others to pump prime. You know. One sort of shows by example in a sense. Somebody has to do it though.
Well, I have a question, but it's actually not to Rudy. Maybe I'm impertinent here. It's to Bertrand. I guess you cannot answer because your lips are sealed. Why on earth did it take more than 30 years for Rudy to get that prize?
Very quick compared to many other prizes.
Well. I guess it had to be a photosynthesis guy to be on the committee.
That will be revealed in 50 years.
I know. Yeah.
You have to be patient.
No, I don't.
I will know.
I don't know if that'll provide an answer, because another person who should have got the prize, probably, many people think so, is G.N. Lewis, who's at Berkeley. And he never got it, but I believe somebody had seen the records then, because 50 years had expired, and there wasn't much information there. I mean, it told you who nominated whom and things like that. But there, as I remember, in what was described in this book, there weren't the detailed deliberations that went on. So you may not, even if you live 50 years more, but now it's just 30 years more, you may not learn. You better find something else to do.
Okay. So with that, we thank Rudy, and we just have a few minutes after we have thanked him for the closing. So thank you again, Rudy, for this wonderful talk.
Okay, thank you. Thank you.